Alright, so in transcription and translation, essentially what happens is inside of the nucleus you have the strand of DNA that is partially broken open, and then a messenger RNA is created. mRNA is created, and so it will have nitrogenous bases, and so they'll either be labeled G, C, A, or T. So we'll give this one a G, C, and A. So after that mRNA is broken up, it will travel outside of the nucleus into the cytoplasm, where the mRNA a ribosome will attach to the mRNA. So we said that this was G, C, A. However, that was just the DNA. Now that it's RNA, the T no longer exists and it becomes a U. So because we said the DNA was GCA, the mRNA would be its opposite. So instead of it being GCA, it would become, the mRNA would be labeled as CGU, because the T no longer, it switches to a U. After that, what's called an would be referred to as your codon. After that, after the ribosome grabs a hold of it, your anticodon. So then this tRNA has anticodons on it. So because our mRNA was CGU, it would be GCA. So after that connects to our messenger RNA right here, the amino acid would drop off and start to build your protein, making the polypeptide.
DNA replication is the process where DNA makes extra copies of itself. Helicase unzips the DNA so it can make another copy. The top of the DNA goes 5' to 3'. The bottom is opposite. Primase functions by synthesizing short RNA sequences that are complementary to the single-strand DNA. Polymers form new copies of the DNA. Ligase forms the two DNA fragments back together.
In the strawberry DNA lab, we mix salt and water together to break open the cell membrane from the lipid, and then we used ethanol to bring the DNA to the surface. Here is an example of us mixing the salt and water together. A restriction enzyme, or restriction endonuclease, is an enzyme that cleaves DNA into fragments at or near specific recognition sites. They are normally used for DNA modification in laboratories, and they are also a vital tool in molecular cloning. Hydrogen bonds are the very strong covalent bonds within the restriction enzymes, while weaker hydrogen bonds maintain their interactions. These enzymes bind to a specific double-stranded DNA sequence and catalyze hydrolysis of phosphodiester bonds in both DNA strands. This happens within or near the specific sequence. Most restriction enzymes recognize sequences of 4 to 8 base pairs. DNA ligases are used to join the fragments of DNA generated by restriction enzymes. Also, enzymes that cleave the phosphodiester bonds of DNA are called deoxyribonucleases, and enzymes that cleave the phosphodiester bonds of RNA are called ribonucleases.
As I said before, most restriction enzymes recognize sequences of 4 to 8 base pairs. As shown here, person 1 has 40 base pairs, while person 2 only has 10 base pairs.
Polymerase chain reaction is when new copies of DNA are made to be reproduced and replenished for other cells. The new copies of DNA are called amplified DNA.
DNA gel electrophoresis is a method used to separate mixtures of DNA, RNA, or proteins according to their size. During the process, the molecules to be separated are pushed by an electric field through a gel that contains small pores.